So we have seen pairwise alignment where we align two sequences. That's called as pairwise alignment or pair alignment. Now we will look into multiple sequence alignment. So that is the alignment of more than two sequences. It's commonly known as MSA, abbreviated for multiple sequence alignment. The most successful alignment is the one that most closely represents the evolutionary history of those sequences. So while we are doing this MSA, we'll incorporate the phylogenetic trees also. We'll cover the phylogenetic algorithms in the later on sections. Here is the alignment, just a view coming out from a software. How does it look when you have multiple sequences? Now each alignment position looks like a column. So we'll start using the words column. When we say column, that means each position. So for example, at column number 1, we have lot of n's and there is one s. And in the end, we will come to a consensus sequence. So what we have, so most of the time we have n. So we can say with confidence that at this position we have n. Then whether those sequences are conserved or not conserved, here we see that there is just one mismatch, so that was not conserved up to 100%. And these bars they can give us the idea of that percentage of conservation. So this is just one output coming from a software. The purpose was just to elaborate that how does it look when we do this multiple sequence alignment. The purpose is obviously to have a phylogenetic analysis so as to construct the evolutionary trees. We can demonstrate the homology within multi-gene families within themselves, as well as if we have some new sequenced genes, we can also incorporate into those gene families. We can use it while determining the consensus sequence. We can identify the functional sites. We can identify the motifs. So sometimes we have on those sequences, there are some areas which are highly conserved, so we can call them as patterns or motifs. We can also use this multiple sequence alignment while we do those protein structure predictions. Here in this example, we see that we have 4 sequences, N, F, L, and S. So the first sequence, we have sequence B, N, F, and S, and they are aligned this way. So if we reconstruct their phylogenetic tree, whenever we are doing those trees, we call the word reconstruct. So actually we believe that these trees, they exist in nature, so we are kind of reconstructing them. So rather than using the word tree construction, mostly we use the word tree reconstruction. So position number 1 is highly conserved, so we have n's all over in that alignment, and this is the finest alignment where we have gaps on all of them except sequence C. Then those A, B, and C, they differ with this one change in that case where we have AB has F's, whereas CDs they have Y's. Same way we can observe one gap in case of sequence B, and again the last amino acid is conserved, so we have all S's. When we put them together in a phylogenetic tree, so here we start, and then we can see on this side we have Y's, on this side we have F's. So we make 2 groups. So these top 2, they are clustered together, and bottom 2 are clustered together. Same way within those trees, again we observe the branches as those sequences, which are actually, we call them as leaves. We'll talk about this phenomenon when we talk about phylogenetic trees. So the leaves are all the individual sequences, and then we have these ones, they are connected over the tree. So here we can have their differences. This guy does not have K, while on this side we have K. Same way if we compare these two, we don't have L on this location. So this is how we can observe their insertions or deletions, as well as the differences between what we can call them as these clades in this phylogenetic tree. So the ultimate goal of doing this multiple sequence alignment is to make some trees like this and to infer those phylogenetic relationships. So this multiple sequence alignment is the alignment of more than 2 biological sequences. These sequences can be proteins or DNA. It is commonly known as MSA.